Right. A lot of people here yet, but typically we get some starters and we get a very good number of people on Zoom. So. Welcome everybody to Pediatric Grand Rounds. Dr. Hirsch is on his way, but because he's a little delayed, I have the honor of doing introductions today. My name is Sandrine van Schaik. I'm the Vice Chair for Education in the Department of Pediatrics. And it's a great pleasure to introduce today's Grand Round speaker, Dr. Apoena Ribeiro. She's the Chief of Pediatric Dentistry here at UCSF. And if you have not heard about her, we won't blame you because she's been here for all of 4 months, joining us from the University of North Carolina. So we're very happy that she's joined her. Dr. Ribeiro is a very unique clinician-scientist, who combines her training as a pediatric dentist with her expertise as a microbiologist. She currently leads the oral health research agenda for the largest cohort in HIV research in the United States and uses advanced omics to investigate how the oral microbiome drives disease in vulnerable populations, including children with chronic illnesses. And today she will discuss the critical link between oral microbial ecosystems and systemic health. And so please join me in welcoming Dr. Apoena Ribeiro.
Thank you. Thank you so much for the kind introduction. And I'd like to start by thanking the invitation from the pediatrics department at UCSF. It's been a pleasure. My last 4 months, a huge change, but also a pleasure to become part of UCSF family and come to the other side of the coast of US. So today I'm going to share with you some of our discoveries and my interests, and also maybe open the doors for continuing collaboration with UCSF and also the Department of Pediatrics. So a little bit about me. I was born and raised in Rio de Janeiro, Brazil, where I got all my degrees and certifications. I am a dentist by training and a pediatric dentist. And then I started teaching in academia and doing some research and also having a part-time private practice. But curiosity was always something that drove me from my career. So I decided to do PhD in science with the focus on microbiology and immunology. So I have this dual training of, and which made me a clinician-scientist by joining both sides of clinical and foundational science. So that's what drives me in my career and how I try to navigate by seeing patients, being in the clinic, having my questions, and then trying to combine clinical research with basic sciences research that I like to call, because it's not basic, I like to call it foundational sciences. My main 2 areas of research are the oral microbiome and its metabolome profiles as determinants of oral health and systemic health, most focusing on children and adolescents. And on the most dentist side, I like to investigate caries diagnosis, detection, and minimally invasive management, so on the prevention side. So today, I'm going to be focusing on the first topic, that is the oral microbiome and how it interacts with oral and systemic health. So today's objectives will be to define the composition and functional aspects of the oral microbiome and explain its role in the initiation and progression of oral diseases in children. Describe how oral microbial ecosystems are influenced by local and systemic factors and evaluate the potential impact on systemic health. and to discuss the importance of oral microbiome research and targeted management strategies for improving both oral and systemic health outcomes in pediatric populations. So, diving into the first topic about composition and functional aspects of the oral microbiome and how it plays an important role in the initiation and progression of oral diseases in children. The latest data from the NIH Human Microbiome Project showed that our human body is made by hosts actually 100 trillion bacterial cells as opposed to 10 trillion human cells that makes up our body itself. So we are big hosts of this whole microbiome. And if we compare the distribution of this microbiome across all the sites, the oral cavity is the second most colonized site in the whole body, compared to the GI tract, which is very big and very long, right? The oral cavity harbors a huge amount of bacterial cells. If we compare the genome, the human genome is around 23,000 genes compared to the human microbiome with more than 1 million genes. So for sure, it has an impact on our homeostasis, on our health and disease. And this is what really triggers my curiosity, is to understand how it is, what is the impact and how we can manage this impact for better outcomes in the clinical practice. And as a dentist, I have to say that I chose to work on a very unique ecosystem in a very unique place, because the oral cavity has a wide range of different habitats with unique environmental conditions. And these conditions will dictate who will be able to colonize and what they will be able to do in those environments. We have multiple types of epithelium: stratified squamous epithelium, keratinized, non-keratinized, gingival tissues, and continuous exudate being produced and released through the crevicular fluid from the gingival crevice. We have saliva, we have gingival sulcus, tongues, cheeks, hard and soft palate, tonsils, and teeth. Each of these areas functions as a small environment in this big environment that would be the oral cavity. So on the tooth, whatever is attached to the tooth needs to be removed. It doesn't detach because the tooth is the only surface and the only tissue in the whole body that doesn't shed. So whatever is attached to it, it will maintain there unless something, a force is put to be removing that microbiome. So it has an impact not only on the tooth level, but also on the gingival level and also on the systemic level. And depending on the site, because of this uniqueness that these sites have, they also dictate what is the composition of the microbiome. So data from the Human Oral Microbiome Database published in 2009, 2010 with the techniques available at the time, they were able to identify more than 600 species, different species from the oral cavity, 35% of which were never been studied before because they could not be cultured. Now we are close to 800 different species colonizing the oral cavity. And if you look on the plot on the right, depending on where you collect your sample, the community is completely different. So for us as dentists, we have to be, or researchers in the oral cavity, we have to be very mindful of what is the question and where do we need to collect those samples. Is it a question related to the disease itself, localized on a tooth surface or the gingival fluid or the gingival tissue? So I need to be collecting samples specific from that site. Or if it's, I'm interested in traveling microbiome and using saliva to travel to other sites of the body, then I will collect saliva. If it's stimulated or non-stimulated, it will also bring different composition in this microbiome. Even if we think about a single tooth surface, depending on where you collect the sample, the composition of that microbiome will be different. So if the sample is collected from the buccal site that we see when we smile, it has lots of Streptococcus genus, but if we collect from the lingual side, only, I don't know if it's showing here, but only in the canine has a lot of Streptococcus. So again, we have to be mindful where we collect these samples. And how do we live in this unique environment with all these changes depending on where you collect, and we live in homeostasis? So the oral microbiota starts to be developed before the child is even born. So we divide the oral microbiome into three main stages: predental, primary dentition, and permanent dentition. Each stage has a unique composition of the oral microbiome, and it will change across the lifespan. The womb of the fetus isn't sterile, so there is maternal transfer of microbes to the fetus that occur via uterine cervix and placenta. So important bacterial samples were isolated from the umbilical cord blood samples originated from the oral cavity. And from amniotic fluids, Porphyromonas gingivalis, that is the main pathogen of periodontal disease. So mothers with periodontal disease, they can have this microbe transfer through amniotic fluids to the babies. What is the impact on this oral microbiome later in the lifespan of that child? So these are questions that we are still investigating. And I will talk a little bit later, but just as a hint, mothers with periodontal disease, the ones that you have gingival bleeding, loss of attachment of the tooth to the bone, they have been highly associated with preterm and low birth with the babies. So it is important to be investigating this and detecting the sooner the better. This is a very interesting paper published in 2011 showing that by the age of 3 months, it is possible to differentiate which baby was delivered by vaginal delivery as opposed to C-section, just by collecting saliva, because there are unique microbes that can only be seen among children delivered by C-section, as opposed to microbes that are only be seen by vaginal delivery. So the mode of delivery also prints an important component on how this oral microbiome will be developed. And these bacteria, this species that I highlighted on the top is Escherichia exigua, for example, that can only be seen in the oral cavity of baby delivered by C-section, is highly associated with aggressive periodontitis in the adult life. So the mode of delivery, the mode of breastfeeding, environmental changes will play an important factor on how that child will develop and have good outcomes at a later life. After birth, the transmission of bacteria comes from the environment, and the initial colonizers will then set up the pathway for the next colonizers to come. So that's why it's so important to really understand how the microbiome on the first days of life will impact the outcome at a later age. In the first few months of life, that baby will receive bacteria from person-to-person transmission, depending on the composition of the infant's saliva, mode of feeding, and microbial crosstalk. So there is an impact of especially the caregiver on the oral health of that caregiver, what is the transmission of those bacteria, because they will set up the pathway for the colonization of that microbiome. To a point that around 5 months of age, you have established already the predental microbiome core, and it is completely different from the mother's microbiome or the caregiver microbiome, because that baby will receive samples from the environment, from whoever is taking care of that baby. The microbiota mostly consists of bacteria from 6 phyla: firmicutes, proteobacteria, actinobacteria, bacteroidetes, fusobacteria, and spirochetes. And these 6 phyla will be maintained for the rest of their lives. They will not change anymore. Most prevalent genera: Streptococcus, Actinomyces, Haemophilus, Neisseria, and Veillonella. Again, the top 5 prevalent genera that will follow you up until you die. Then we have a big event happening in that oral cavity, that is the first eruption of the first tooth. So then it's when it comes the first surface that doesn't shed. And it brings a huge change in that environment, because for the body standpoint, whatever is attached there will stay, will stick, and the body will understand that there is an infection coming. So that's why babies will increase salivary flow, will be irritated. Some of them will develop rash as an immune response to that change in the composition of the microbiome. Also, with the eruption of the first tooth, you will then have gingival sulcus and occlusal surfaces, which will bring for the first time areas with low oxygen, allowing for anaerobes to be able to survive, and the crevicular fluid that will bring lots of IgG and IgA to the environment. So once the first eruption happens, then you start to develop your core microbiome. And this core microbiome will be stable throughout the primary dentition up to your permanent dentition. And by the age of 5, whatever is established as your core microbiome will follow you until you die. Of course, changes because of systemic diseases, use of antibiotics will make changes, important changes to this microbiome. But when you stop or you end with the antibiotic taking, for example, your core microbiome will come back. There's a memory of or a fingerprint of your oral cavity microbiome. And this core microbiome has lots of benefits, and we call them resident microbiota. It is part of the development of the host. It's part of the innate defense system by causing a saturation of microbial adhesion sites that will minimize and form a barrier against transient microorganisms, not allowing these transient microorganisms to establish. They will also act for competition for essential nutrients and create a condition that is not favorable of the growth of transient microorganisms, and also produce lots of inhibiting factors to maintain this homeostasis. So our core microbiome is essential to maintain the homeostasis of the oral cavity. The main problem is when we have a shift on this balance, allowing that the same core microbiome can then become the pathogens or pathobionts and develop the oral diseases. So when this equilibrium shifts to a dysbiotic state, that's when the problem starts. So what would be the microbiome dysbiosis associated with the oral diseases? And why it's important. So oral diseases are affecting now, according to the latest WHO data report, 3.5 billion people worldwide. And dental caries is the most prevalent chronic disease in the world. It's not diabetes, it's not hypertension, it's dental caries, affecting 2.5 billion people worldwide, with a 15% increase in disease over the last 10 years. So as a dentist and caryologist, I'm deeply, deeply affected by this data. Periodontitis or periodontal disease affects 19% of the global adult population with more than 1 billion cases worldwide. And what is interesting to note is that both diseases, they are caused by core microbiome that once were in homeostasis, but changes in systemic health or local behaviors will give them the tools to become pathogens. So how do we define dental caries, the most prevalent disease in the world? It is a polymicrobial biofilm mediated disease associated with biochemical imbalance between the tooth and the biofilm. Remember that it doesn't shed. So if you don't remove that biofilm will maturate and that bacteria will be able to produce acid, causing a biochemical imbalance between the interface of tooth and biofilm. And these acids will allow for demineralization of the tooth structure. That is what we call the initiation of the disease. This demineralization is dependent on fermentable carbohydrates. So sugars and all fermentable carbohydrates that we intake in the diet are also the fuel for these bacteria. In terms of a global prevalence, I'm bringing here a map to see where we are in the US for early childhood caries, which means children under age of 3 affected by this disease. And as you can see, US here on top. I don't know where oh, here I am. No, it's not showing. But anyway, US accounts for 26 to 50% of the children under age 3 with the disease. My home country, Brazil, between 0 to 25%. So I think it's one of the only statistics that I'm better, that Brazil is better than US. But anyway, we need to do something because the knowledge is here, the opportunities are here, but we're not doing a good job. And this data is considering early childhood caries only when the tooth has already a cavity, a hole. It's not considering when the tooth is not cavitated yet. So previous publications showed that these kids, they have a high prevalence of the common bacteria that everyone has, but they have very active species causing these diseases. This is a picture of one of my patients. She's 2.5 years. I had to go to OR, of course, to treat her. And front teeth had to be extracted. lateral teeth I could restore, lots of root canals on the back at the year 2. So also, kids that have the risk but don't develop the disease, they have a unique microbiota. And I'm going to show you some of the data that we found in a minute. So this is a cohort that I had the privilege of being the pediatric dentist and microbiologist for the team. And the PI is Dr. Kimon Divaris, far left, from UNC. And it's a U grant that we got the opportunity of mapping early childhood caries across the whole state of North Carolina. So we evaluated close to 7,000 kids between 3 years old and 5 years old. And the prevalence in North Carolina was 92% of the kids at this age had at least one caries lesion. We then randomly selected 300 samples to do whole genome sequence because I was interested in seeing if with this new technology that we can go deeper into species level and understand what they do with metagenome analysis. We sequenced in my former lab at UNC 300 samples, 150 from kids with caries and 150 without caries. And from the results, we could identify any publishing Nature, the PMID number is right here if you would be interested in reading. We identified 12 players that were highly associated with caries disease among these children. Among these 12 species, we selected 5 species that I could order ATCC samples and test in the lab. What are they doing that make them so unique among this population? And we saw that when paired together, these species with the main classic bacterial species associated with caries that is called Streptococcus mutans. When you pair Streptococcus mutans with these species, they produce such a thick biofilm that it's so hard to be removed from the tooth. And it's very sticky and hydrophobic. So it creates a barrier in a way that toothpaste doesn't penetrate that biofilm. Fluoride doesn't penetrate in that biofilm. So bacteria gets protected and continue to produce acid unless you really remove that biofilm. We then tested in animal models using rats in my lab, and we saw that when we paired them, this was the severity of the lesions when you only had the classic bacteria, Streptococcus mutans, inoculated to those rats. But when you pair them with these species, the severity of the lesions was like highly increased. So they work very well together. So we started to then think about how can we apply this to the clinic? Could we differentiate this biofilm? Could we develop a chairside strategy to identify if that child has these bacteria active so we can improve orientation and prevention methods? So that's where we are going now with the renewal, hopefully, of this grant. So for the first time, we showed that strep mutants, the classic one, interacts with the other species, Leptotrichia wadi, Prevotella salivae, and especially Selenomonas sputigena, to enhance the karyogenicity or the ability of that biofilm to develop the disease. On the other side, from those kids that besides all the odds of having the disease, besides everything that behavioral, sugar intake that they were exposed, they don't have the disease. Turns out that the unique species that was present in those kids and was not present with the other group was Streptococcus oligofermentans. So from the metagenomic analysis, we then did the same thing. Let's bring to the lab and see what it does in the biofilm. So we tested in these spheres of enamel. And we saw that when you put this bacteria with the classic one, Streptococcus mutans, turns out that it produces hydrogen peroxide and it kills the Streptococcus mutans. But those that are able to survive, and we knocked out the gene for hydrogen peroxide production. And even though without the production, turns out that it produces something that we don't know yet. We are doing the sequencing analysis that changes the behavior of mutants, and it doesn't allow it to attach to the enamel anymore. It drops on the tube. It doesn't attach to the enamel. So something is this bacteria doing that is causing this change. So again, thinking how we can apply it, because that's how I drive my research portfolio. I always need to bring to the clinic, right? What can I do to improve my treatment? So maybe this can be a good probiotic that can be, since it is a commensal bacteria, it can be given to the baby at an early age, enhancing colonization of these bacteria to enhance competition. So that's where we are moving with this study. We were also interested in understanding if the microbiome changes once you become a scholar, after 6 years of age to 12 years of age, while your tooth is erupting. The main problem is that all teeth, when it starts to erupt, it still needs 20% of the calcification that is done across 2 years with interactions with saliva. So these teeth are very much at risk of developing diseases because it has large pores. So we wanted to see if these large pores and these teeth, they are also at higher risk for having a unique microbiome attached to it. And we could identify the set of these species on top of the graph and were associated with health, and the ones with red are associated with higher risk of developing the disease. We followed these teeth for 1 year to see if those with this composition of the microbiome would actually develop disease or any lesion in 1 year. We also saw that, of course, the microbiome will be different if the surface or the tooth is sound compared with the teeth that have disease that we call active white spot lesions. So the initial demineralization, it's not even cavitated. So we diagnose before cavitation because then you can do minimally invasive. You don't need to do surgical intervention on those teeth. But we also saw that the composition of the microbiome completely changes depending on the amount of sugar that you intake across the day, especially in between meals. So if we want to gather information from the patient or from the parents or caregivers, the question is not if you take sucrose or if you take candies, but how many times across the day you take candies, because it will influence the composition of your microbiota, selecting for a more pathogenic microbiota, especially in between meals, because once you eat something with sucrose, it takes 30 to 45 minutes for your oral cavity to return the pH of the oral cavity to normal pH, around 6.5 to 7. So for 30 to 45 minutes, your oral cavity will be at an acidic pH because you ate sugar. So imagine those kids that at every time running around and they get candy, and then 30 minutes later they get a candy again, and then they get a candy again. So it is the frequency and the intervals between the meals that will dictate the change in your oral microbiome. We also wanted to understand if after we treat those patients, if the microbiota will also be stable or back to a normal and healthy microbiota. Or once you have the disease, you always have a higher risk of developing new diseases, new lesions. So we collected samples 1 year after we performed the full treatment of those kids, and without any instructions to go home or changing the diet or change. Well, we gave the instructions. You need to change the diet. You need to improve your brushing, but we didn't recall to reinforce. We didn't apply fluorides for 1 year. And so 1 year later, when we called those kids again, we could see that the microbiome was completely stable once you bring back health, meaning that it's not a stamp on your passport. You had caries, you're gonna have caries the whole life. These were the species that were decreased on the left and the ones that were statistically significant increase, meaning core microbiome associated with health. And among the Streptococcus, we saw an important decrease of Streptococcus mutans, that is the main pathogen one, even after 1 year being collected. We are also interested to see if it's only bacteria associated with these diseases, or if we have interrelation interkingdoms. And we started to investigate the role of Candida across those kids with early childhood caries. And turns out that Candida is really a good partner with the cariogenic bacteria. It forms a mesh that increases the attachment of that biofilm to the tooth. So why is it important? First, because those kids that have any kind of immune suppression or before the first 6 months of age that have candidiasis, they have a higher risk of maintaining Candida, especially Candida albicans, after the core microbiome. Together with these bacteria that are highly cariogenic, they will form a protective mesh, making them highly attached to the enamel. And also, the byproduct stimulate the activity of these bacteria in producing more and more acid. So in other words, we have to also be paying attention to those kids that have any kind of immune suppression because they are at higher risk of developing the disease. So how these oral I've been talking just in relation to oral cavity, but how does it can influence systemic health and are influenced by systemic factors? My two main areas of interest are respiratory infections and immune suppression. So, but there are plenty publications in the literature showing that the oral microbiome have a huge impact and have been associated with a higher risk of stroke, Alzheimer's disease, heart disease, uncontrolled diabetes, osteopenia and rheumatoid arthritis, cancer, and preterm and low birth weight babies. So the impact, what happens in the oral cavity doesn't stay on the oral cavity. It's different from LA, right? Or Vegas, I mean. What happens in Vegas stays in Vegas. Here, what happens in the oral cavity doesn't stay in the oral cavity. It affects the whole body. So my two main research focus on the respiratory infections. I've been working with patients with cystic fibrosis and patients with asthma, kids with asthma. And for immune suppression, kids living with HIV and women at reproductive age living with HIV, because I'm interested in preterm and low birth weight babies. So some results from our project on cystic fibrosis. We are now, this data is not published yet, and it's across 104 kids and adults living with cystic fibrosis. But we are now in 211 participants. This was a poster that we presented in the beginning of this year. And we can see that adults are more affected by oral diseases than kids. But what really struck us was the high prevalence of tooth defects. Primary tooth or baby tooth, they are formed intrautero. So whatever happens during pregnancy will affect baby teeth formation. After birth, whatever happens to that kid will affect permanent teeth and can cause defects that will increase the risk for oral disease, especially dental caries. And these patients, they have a very high prevalence of enamel defects. And we also saw that FEV values are correlated with a lower diversity of microbiome in saliva. Lower diversity means higher risk or risk of dysbiosis, because we want a highly diverse microbiota at a low abundance of each of these species. So the lower diversity in the oral cavity means that there is a shift to dysbiosis already. And we also saw that in adults, lower FEV, lower lung function was associated with patients with dental caries. In terms of microbiome composition, it was very interesting to see that those with poor lung function measured by FEV1 and FVC ratio, they were the ones with higher prevalence in the sputum of bacteria from the same genetic background from the oral cavity. And I highlight here Treponema, Pseudomonas, Emojibacterium. So we think that these bacteria in saliva, they travel to the lungs, and it's an important source of pathogens to the lungs and can be associated with poor lung function, even if these kids, they are under CFTR modulators. We also saw that the composition of this sputum is totally different if the patient has cavities on the tooth. And those patients with chronic antibiotics, they have a lower diversity in saliva of the microbiome, meaning there is a dysbiosis, and a lower composition of this community as well in B. And here I list the most prevalent ones. So the oral cavity can have an impact of the lung outcomes of these patients, and we are digging into more analysis to see how it is impacted. For sure, we are now comparing composition of saliva of plaque, biofilm, and the sputum to see if they match. For the HIV cohort study, I have the honor, the pleasure of being part of the MWCCS, that is the Max-Wise Combined Cohort Study. The main center is here, UCSF. And the aim is to characterize the aim of my agenda is to characterize the oral disease among MWCCS participants and compare with adults not living with HIV. And also, part of this project, as a parallel project, is the STAR, that is the study of treatment and reproductive outcomes. So women at reproductive age living with HIV. So we enrolled 526 participants and examined these participants, and we could see that 2.3% of these women at reproductive age, so at between 18 and 45 years old, they have 2.3% of edentulism, so no teeth already. And the national prevalence is 0% by the age of between 18, 24, 0.2 by the age of 25, 34, and 1.1% by the age 35, 44. So oral health among these women is really, really bad. And we found 35% had some oral findings that I will share with you. Soft tissues, we were able to identify 19% of the patients with oral candidiasis, 0.5% with hairy leukoplakia, 1% herpes, some brownish red lesions that were referred for histopathology, and inflammation in the gums, 16%. And hard tissues, 27% of the patients had open cavities like this one, and 24% had only roots left in the oral cavity at this young age. So what we are doing now, what we submitted, and we got scored, but we didn't get funded, is that these women, some of them are pregnant. So I'm interested to see how this oral health will impact the oral health development of these babies when they deliver. So babies exposed to HIV. Hopefully we'll be resubmitting it in the next window. My background with pediatrics and HIV came from Brazil. That was my master thesis and my PhD thesis, where I developed an oral health program specifically to HIV, kids living with HIV back then in Brazil, after I saw that many of them had many oral lesions, lots of caries activity, like 8 teeth in medium with active lesions. So the disease was progressing. And a high association with these lesions with plaque accumulation. So I really needed to do something for these kids. So we developed this oral health program at 2 public hospitals in Rio de Janeiro, free of charge, only for with donations, and we were able to provide care to these kids. So what is the importance of oral microbiome research and targeted management strategies to improve both oral and systemic health? This was a recent publication that we did, where I was constantly asking, okay, we know all these species, we know the relative abundances, they are associated with health or disease, but what can we do to the patient? So we published this review article, and surprisingly, it's one of the most cited article in this journal in the last 5 years. And I was like, what? Okay, so people are asking the same questions. What do we do next? So some considerations, we need to enhance population-based longitudinal studies to better understand the role of this oral microbiome and the impact on systemic health. and consider application this knowledge to precision diagnosis and management. We cannot continue to use a cook recipe for everyone. We are individualized patients, people. We need to move forward to precision and individualized treatment. Translate this knowledge to clinical settings by improving risk management, disease prediction, and preparing clinical guidelines to identify susceptible individuals, so then you can make the impact and change before the disease starts. And improvement on prevention and treatment methods. So some of the ongoing projects, and this is an open door and invitation for those who like the topic and would love to collaborate. I'm always collaborating with multiple researchers. And so as I mentioned before, we are now resubmitting the R21 that we didn't get funded to investigate the oral microbiome development of those infants living with HIV, perinatal exposure from those women from our cohort from STAR. STAR is across 6 sites in the US. It's not in California, it's in the South. But max-wise, the main center is here at UCSF. So I'm including UCSF as one of the centers to continue investigating, or to start investigating the oral microbiome development and follow up these kids to see the impact on their oral and systemic health. The other project that we are submitting in cycle 2 is what we saw with the cystic fibrosis, which was very interesting, was that we could identify 3 main different sets of bacteria that or clusters that we called Oro1, that is associated with patients without disease, Oro2, patients associated with oral disease, and pathogen, those patients with low lung function. And we saw that patients from oral 1 would go, so health would go to oral 2 and come back with a very minimal shift to pathology. But those patients with oral disease, they have a shift and are highly associated with those pathogens, those clusters from patients that have low lung function. And I saw that the saliva has something different. And I started to dig deeper, and turns out that these patients, they have defect on the CFTR protein, transmembrane protein, that changes the concentration of chloride ion, bicarbonate, thiocyanate, and sodium. Because I was very intrigued, because the microbiome of these patients is so unique, the core microbiome, so different from normal microbiome, that I said, there must be something, not only medication, because that is causing it. And so we are now willing to investigate if these changes in these ions are determinants for the microbiota. For sure, sodium will play a big role, because these patients, they have a high amount of Staphylococcus aureus in the oral cavity, which is not the regular site to be found. And Staph aureus loves a high concentration of sodium. So then my brain starts like, okay, in the early ages, the main pathogen on the lungs for cystic fibrosis is Staphylococcus aureus. Maybe we're selecting Staphylococcus aureus because of high concentration of sodium in saliva. What we can do? So this is what we are planning to investigate. And on a side project, I was asked a question from a pediatrician at UNC that, came to a talk and said, my patients, my adolescents that I'm treating for opioid use disorder, once I start treating them with buprenorphine, that is the SOS drug, the oral health declines abruptly in 6 months. They start to show cavities. Some of them start to show abscesses. Something is happening. So I was like, okay, let's start to dig into it. And we already submitted a paper for publication that turns out that, in fact, buprenorphine will alter the microbiome and make them so active and producing the EPS, that is the glue to attach to the enamel. So they produce thicker biofilm, again, protected from saliva, from fluoride from toothpaste, from water. So it's very, very interesting, the impact of this medication in the oral cavity. So we are planning to submit RO3 to continue investigating this with some samples that we want to collect from these patients. So in summary, I don't have my time. I don't know if I'm already over it. But in summary, I hope that I could show that main oral diseases are caused by a dysbiosis of the core oral microbiome, that the composition of the oral microbiome is impacted by local, systemic, and environmental factors, which in conjunction will dictate the risk and development of oral diseases in children, and ultimately advancing oral microbiome research and developing targeted management strategies is not just an oral health priority. It is a powerful pathway to improving lifelong systemic health for every child. So I would like to enhance the recognition and modulation of the oral microbiome because it represents a critical frontier in pediatric care, with the potential to measurably enhance both oral and systemic health outcomes. And with this, I say thank you so much for staying with me. And this was my welcome party at the Orofacial Sciences Department. I feel so welcomed here and at the School of Dentistry that I couldn't choose another picture different than this one. So thank you so much. Here I have some faculty, my residents, some of my lab team that I brought from UNC. We are a family, and I can take any questions if you have. Thank you.
I have one question here from the Zoom. You actually have a fairly robust Zoom audience, in case you were wondering where everybody is. Not everybody made it out in the call today, but there are on Zoom. There's a question here that says, thank you for this wonderful lecture. You mentioned the frequency of sugar consumption changing the oral microbiome. But what about carbohydrate-rich foods like crackers, goldfish, and muffins, which are offered very frequently in schools and after-school programs here?
That's an excellent question. Turns out that they also have a high content of sugar. So it is the sucrose that is the main villain. Not saying that the other sugars cannot be metabolized, but these cardiogenic bacteria, they love sucrose because with the enzymes that they produce, they will break down sucrose and form the EPS, the glue. And also, once they are attached to the enamel, they will continue to intake sucrose because they are fantastic. I love them. Well, I don't love them, but they are fantastic because they have a pantry inside of them. They accumulate inside vesicles internal polysaccharides to be used in between meals as a source of energy. And with the metabolism, we'll also release acid. The end product of the metabolism of the sucrose is always acid lactic. So meaning that what you ate now will be eaten by the bacteria. They will put and store inside their pantry, inside of them in vesicles. And then when you're not eating and they are fasting, they will use those internal polysaccharides to continue to survive. So the main problem is the sugar hidden in these crackers. So, and when I say in between meals, we saw that the main threshold is 5 times during the day. So what I do, I didn't know if I would have time. So what I do with my kids in the clinic, I suggest parents to prepare a box, and I say that it's a treasure box. That treasure box will be decorated, will be like crafted by the kids, like let's do it, a project for the weekend. And that treasure box will be filled with candies during the week. And then that box can only be opened on Saturday or Sunday. On Saturday and Sunday, let them eat everything at once. Before going to bed, brush their teeth. And then close that box, fill it again, full of trash, and open again once a week. And it's that treasure. So, because I know grandparents, they like to bring some candy. So that's what I tell the parents. So they understand that it's the frequency and also consistency of the sugar is very important, because the more sticky, it will stay forever.
Well, we're over time. Thank you so much.
Thank you. Yes. So I'm leaving my email. So if the Zoom would like to send me the questions, I'll be happy to answer. And thank you so much for your attention. Thank you.